So we'll start in just a moment. We're just waiting on Dr. Peterson to come back. I was going to talk about a little bit more about SP at the end, but I'll do it now just to save on time. So I just wanted to thank CIHR in general for helping to fund this forum and the last one that we did. If you weren't present at the previous forum, if you want to watch it again, if you look on our YouTube channel or you go on our website, you can look at what we had done previously on personal genetic testing. There's also something that's being done by the FRQ, the Fonds de Recherche du Québec. They did a public call for their essentially for their vision for the next few years of what they should be working on. They want to know what the public thinks of this, or what the public thinks that, what kind of research should be done in Quebec. So if you wanted to go to touspourlarecherche.com, you can actually vote for your favorite ideas, or you can submit your own ideas. So the chief scientist is very interested in finding out your thoughts about this. And also SPE will be at the Canadian Science Policy Conference this year in November. We'll be gathering media strategists as well as science communicators, people that have been very successful in reaching out to the public to really see how we can establish a better dialogue between scientists and the public like you. So if you have any particular ideas, if you know of excellent events that have happened that have really engaged you, if you want to let us know, we'd be happy to bring these up in our panel. So without further ado, I'll leave it to Natalie to go through the questions that you've asked.
Terrific, thank you very much. Do we need to speak into these, or no?
Hey.
Yes, okay, terrific. So the first question reminds us that even those of us who think we're making very good efforts at science communication sometimes overlook the most obvious thing, namely, what does CRISPR stand for? None of the three of us shone on explaining that part. Would you like to take that one?
I would, if I could remember. I wrote a very nice review about CRISPR and very nicely spelled out all of what it meant. And almost every paper in the literature says exactly what it is and then puts CRISPR in brackets at the end of it. But you edit over that because you know that you're talking about CRISPR. Somebody in the audience certainly does.
Clustered, regularly interspersed,
yes.
Palindromic repeats. Okay. I knew the words. I don't know what it means. Is there an explanation of what that means, or that
Sure.
probably is a little...
Sure. Remember I mentioned that in 1987 there was this funny sequence discovered, and it was published just because it was a funny sequence. So clustered means that there were several repeats. They were regularly interspersed, so there was this cluster that did not look like the normal sequence of DNA. And that's what its name implies. What it is the locus where it's the place in the bacterial genome where it copies DNA from incoming viruses and puts little pieces into it. So it can be many, many different pieces of Oh, I'm sorry. It's on? Yeah, okay, thank you. So it's the place in the genome of the bacteria where the bacteria inserts copies of the invading virus. And it's small pieces, and they're in sort of tandem arrays. Does that help?
All right, thank you. So we'll have each of our speakers read the questions that were specifically addressed to them. So Erika, would you like to read one?
Okay, so my first question is, why is Canada so restrictive about gene editing? What are the main reasons? So it all goes back to the adoption of the Assisted Human Reproduction Act, and that was during a time where there were a lot of fears about dystopias. So it was around the time of Dolly, so the cloning, there was the Raelian hoax. There were a lot of things that made society as a whole fearful. And so when the policymakers, the regulators actually sat around the table, their goal was to protect. And so they said, well, we don't want this to happen. We don't want humans to be cloned. We're afraid, so we're just going to ban everything and turn them into criminal offenses, which at the time, I guess, might have been justified. But as we can see now, it is hindering the evolution of science.
Would you like to read one?
Okay, so while making one genetic sequence change, could there be unforeseen impacts on other traits? That is a wonderful question. And it's wonderful because we really have no useful answer to it, except for, of course, there could be. I might just take you on a little diversion into mouse genetics because it emphasizes how little we know. If you start off with one pair of mice, male and a female, and have them produce a litter, and you take one female and one male from that litter and mate it together to give rise to the next generation, and you do that for 20 generations, you end up with the workhorse of mouse genetics, which are called inbred strains. They are what we call homozygous at every place in the genome. That is, they don't have any genetic variation that comes from mom and dad. They get identical sequences from both. So there's no lethal genes by definition. So if I do that and I have multiple inbred strains and I start off with one female from one strain and one male from another, you'd think that you could cross them together and you would get out mice at the bottom. And no matter how many generations you go. But the extraordinary thing is if you start off with 100 pairs, you're very lucky if by 20 generations you've got 10 to 15 lines of mice that are still viable. And that speaks to a phenomenon called epistasis, which means that certain genes like to live with others. They're not lethal by themselves, but they are critically important that they have in their neighborhood partners that they like to work with. And so we know almost nothing about that level of genetic information exchange. So certainly, if you modify a gene, you could have consequences on other traits in the organism because you're now changing the environment that other genes in that mouse like to live with.
Terrific. I'll read a question that was posed to all of the speakers, a very important one. Should germline gene editing to eradicate inheritable, life-diminishing diseases be possible one day? How should we go about implementing these alterations to the public? Should they be voluntary, like dental work, or regulated, like vaccines? One thing I'll say is that this question reflects why, at a number of public fora and discussions and meetings on human genome editing, disability activists have been very, very concerned about the potential for just this kind of question, because there's the concern that if it becomes possible to eradicate very severe, sort of catastrophic diseases, there will also be the expectation that not only should we eradicate those, but we should eradicate maybe life-diminishing or different conditions such as dwarfism or Down syndrome or deafness. And in these kinds of areas, disability activists say, you know, this is not something we want. We do not necessarily want the ways in which we are different to be eradicated, partly for reasons of human diversity, partly for reasons of the support that society currently gives to people with disabilities. So it's a really excellent question as to what would be the societal norm and expectation if this technology becomes possible.
So if I may comment on that, I know that was something we've recently looked at, and so as with any new technology, we can presume that when gene editing does become used in the clinic, it will be prohibitively expensive at the beginning, and so there's the issue of access. And one thing that we had proposed was that perhaps there would be criteria that would be placed so that those who would need it most would have access to it. And it would be a matter of gauging that as opposed to just making it available to those who have the means to pay for it.
Did you want to add anything on this?
No, I'll leave that insight to you.
Okay, would you like to go with another question?
Sure. So the next question is, my 2-year-old son has an extremely rare genetic disorder called FOXG1. Is the CRISPR technology something that could work and help edit his gene? Now, I don't know the specifics of that rare disease, but if it were a monogenic disease, it would potentially be one of the easier ones to to correct using CRISPR and gene editing. Also, the whole notion of rare genetic disorders within the discussion of CRISPR is something that has been pushed to the side a bit, and having had the chance to speak to patient advocates and individuals with rare diseases, it's something that they feel is really important and they'd like to have a voice in this discussion. So it is something we do need to look forward to. And then there's also the idea or the concept of, well, if gene editing were to move into the clinic, would it be to cure rare genetic conditions, or would it be to cure something like cancer? And so that's something we need to question and we need to think about.
My understanding is that there are currently many clinical trials ongoing that have been approved. Certain number, as an example, for various types of blindness, where there's relative access to the cells that are affected in the retina. Possibly, I don't know the details of them, but I believe they would be using viral vectors to deliver the CRISPR components to the cells. I couldn't agree more that it's going to be the single gene defects, mutations, otherwise that are going to be the most promising targets for effective therapies. And my own feeling is that there is likely to be, certainly in my expectation, in the next 10 years, I wouldn't be surprised if CRISPR wasn't the therapy of choice for a number of inherited conditions.
Thank you. Do you want to read one of your own questions now?
How could the use of CRISPR to edit germline DNA affect the course of human evolution in the future? So it certainly could, but I think taking you back to the railroad analogy, 3.2 billion base pairs of DNA in each cell is a pretty big target. There is certainly a possibility that one could target a specific gene that you think may have a dramatic effect on human development or growth or size, for example. Editing a receptor for a growth hormone or something to create an enhanced response could change our size. There are ways that attacking very specific genes that we have some familiarity with could certainly have dramatic consequences on individual humans. How we then go about changing the future of our evolution would, I guess, depend upon how we go about those individuals who have changed, whether they're reproductively successful in the society that they find themselves in. But yes, the opportunity for massive changes to an individual with subsequent consequences on their reproductive fitness could certainly be a consideration. Probably not of my generation, but very likely it could happen in yours. But the consequences would probably not be felt for a few millennia.
The next question is, do you believe that CRISPR-related patents harm or stimulate scientific research? So the whole notion of the patent war that's ongoing between the Broad Institute and Jennifer Doudna's institute. I've done some reading on the patent war, but I wouldn't say I'm well versed in it. But my opinion would be that putting CRISPR-related patents into place would do more harm than they would good with regards to scientific research. And if anyone's interested, there is a lawyer in the States whose name is Jacob Shirkov, I believe, and he's been following this debate and writing wonderful articles following the patent debate. So I'd suggest for anyone who's interested, you should have a look at his articles.
Maybe I could just add a comment to that. For some of you who may not appreciate the relationship between patents and academic research, for a laboratory like mine, the role of patents is completely invisible to us. All of the applications that we use CRISPR for are not commercially based. We don't expect to have commercial products from them, and we are totally free to use at will all of the CRISPR reagents that exist in the world.
I will read a question which I'm sure applies to both of our experts here, and that is, how do we organize a serene and informed debate on CRISPR implications when so many educated people are rejecting science, being anti-vaccine, creationism, homeopathy treatments as examples? A really important question. Which of you might like to start discussing that?
I will take a stab, not at answering it, but making a comment. I have complete lack of understanding how a country like the United States could lead the progress of science with research institutes that are at the forefront of so many areas of human discovery, and yet be a nation where more than 30% of the people don't believe in evolution. It is totally unimaginable to me that there could be such a dramatic schism between various populations in a country like that. And I think that does bear a little bit on the question that was being raised.
One thing I'll add, actually, just as a segue to that, is there is a burgeoning field that studies the science of science communication. It's sort of cognitive psychology and sort of behavioral economics, and this field, people are increasingly realizing that the way to get people to engage in what we might think of as a constructive way with science is not to bludgeon them over the head with facts and more facts and more facts and evidence and data. It's to try to understand what kinds of frames and identities shape people's thinking. And as you said, it's in the United States, and let's face it, in Canada too, there are both people whose identities and ways of thinking are very much bound up with rationality and science, and those whose identities and ways of thinking are bound up with religious frameworks, with other kinds of frameworks which lead people to define themselves and their identities in ways that are not so friendly. And understanding how to shift conversations is not a matter necessarily of throwing facts at people, but of framing the issues in a way that will be sympathetic with and fit within people's existing worldviews. So that's actually one. There's a whole realm of scholarship now that's addressing this kind of question.
I don't have anything to add. I think that was quite eloquent.
Okay. Terrific. Another question?
Sure. So this question is more personal with regards to my academic progression. So do you have a scientific educational background, or did you get into it after your legal studies? So my undergrad is in psychology, so not... So I didn't really have a big scientific background, and then I just happened to start working with the center and Professor Bartha Maria Knoppers, and they kind of converted me over to the dark side of law. And so now what I do for a living is I work on policies and regulations, so that's kind of how it flowed, and our center really is focused on genetics and gene editing technologies and various aspects, so that's how I came to be where I am.
So this is, in many ways, somewhat a related question. What's the future career path for a current PhD neuroscientist involved with CRISPR in neurodegenerative diseases? I was going to interpret this a little more broadly than perhaps it was stated, because I think it is a broader question. I mean, as many of you are aware, in Canada, there is current consideration of what's called the Naylor Report, which is a discussion of how science is, how science is organized in Canada. I think we've seen a series of decades where scientific funding has been appropriated for particular purposes. And it all makes such tremendous sense when you make the argument at the time for a particular need. But here we are talking about CRISPR. And CRISPR was discovered by basic scientists stubbing their toe on things that they couldn't understand, not because they were looking for a supernova of gene editing technology. And that's the way it's always worked. There have been a few examples of targeted attempts to develop vaccines for particular diseases, for example, polio. But the vast majority of radical breakthroughs in science, particularly biological science, have come from basic scientists doing things that would have virtually no possibility of being explained as being valuable to their society at the moment. The best I've heard of this came from the popular scientific commentator, his name will come to me in a moment, who said, if you ask somebody, a basic scientist, what they're doing, they'll say, it doesn't really matter, you won't understand, but you'll probably be taxing me on it 20 or 40 years from now. And I think that's right. Obviously, there are many dead ends, but there are many dead ends because we have breathtaking ignorance of our world, and probing things that are of interest that can be places where we can make progress is the best way to move forward. So the Naylor report is an attempt to evaluate Canadian science and see if there can be ways of augmenting basic science set in academic institutions such as McGill. And I think we're all encouraged by the positive things that came out of it, and it would have a particular impact on this specific question that's being asked. I mean, people talk to me often, young people, about what to do about their career, and I say, I can't tell you, because the doors were all open when I came on the stage to look for ways to pursue my interest in science. Their doors are closed, and they have to pry them open. There's no relationship. And if I could leave science knowing that there was opportunities for young people coming along, I'd be a lot happier than I am right now. Right.
Got a great question here for the whole panel. A great question for the whole panel, and one that I think will point out maybe some of the incommensurate worldviews of even people within this room. Namely, why would people say no to using human gene editing for human enhancement? We've been enhancing our life ever since the beginning. So, why would people think there's a problem with this? Someone skeptical that anyone would think there's a problem with it.
Well, the main argument that goes along with this is, oh, well, we're trying to play God, which, when it comes to enhancements, it will typically come up in, let's say, the sports field, or as you had mentioned earlier on, with regards to even soldiers and enhancing soldiers. And I think there are a lot of social and ethical and moral implications to that. We're playing with the essence of what being a human is. I'm not saying that I'm completely opposed to the whole idea or opening up to it, but I think something that's at the heart of it is really considering the safety of human beings. So before we even venture in that, I think we need to make sure that whatever we propose or whatever technique we're going to use to make these enhancements is as safe as it possibly can be. And then once we're there, crossing over into the enhancement realm is crossing a whole other moral line that I think discussions and debates should be had at that time, but it's not something we need to address necessarily right now.
Your thoughts?
It really is the most important question to ask, because I believe we're on the threshold in the next lifetime of having enough insight into biology to be able to do things that could be defined as enhancements. For me, I think I would have to know precisely what was being suggested, and then have an opportunity to really evaluate what the consequences would be. We really do have breathtaking ignorance, and it would be, I think, totally inappropriate to think that we can just grab a CRISPR kit and go ahead and make people who are going to live longer, be smarter, because we don't know how to do that. We're learning at a tremendous rate how to do it, or about what's involved in it, but we don't know how to do it yet.
One further way you could think about this question has to do with the issue of social inequality. The idea that conceivably making enhancements in people would enhance not only one individual person, but the whole line of descendants from that person, and raises the specter of entrenching inequalities in a much more profound and long-lasting way than they already do exist in our society. And given what we know about the inequality of starting points and life opportunities and what that means, would we want to pound the table and say, this is one line we won't cross in terms of adding further inequality? Or would we say, look, inequality is pervasive. Why should we say this technology is somehow special and off the table? So important questions. You want to go for another one?
What's your opinion about the legal issue of slowing down science? So with regards to CRISPR-Cas9. So I had mentioned that in relation to the moratorium that was called for back in December of 2015. I think at the time when CRISPR had kind of hit the ground running and it had come to the forefront, I think people were more worried than anything. So I feel slowing down science at that time with the moratorium was a valid decision. That being said, being a year and a half from that point, I think we do need to start reconsidering that moratorium and seeing, how we can move forward and lifting it and allowing certain applications to move forward. I think, in my personal opinion, germline gene editing for treatment and prevention purposes would be a positive thing. So I don't think we should have this hold back that research aspect of things.
Perfect. And we just have, unfortunately, we have time for just one final question, which I'll pick one, I'll use one as the wrap up to this whole session. Who makes the decision regarding the ethics of gene editing research? So when all these issues have been raised, who decides what research will be done to open all these doors?
I would turn to you.
Do you want to take a stab at that?
Sure, I'll take it first. So I don't think there's any one body or one person that makes this decision. So when we're laying out the ethics or the ethical framework surrounding gene editing, there are multiple stakeholders around the table that have these discussions and put forward statements, guidelines, and regulations. That being said, when it actually comes to what is ethically permissible or not to research, I'll go back to one of the main stakeholders that are missing from the table are really the public. I feel we're hearing from the ethicists, the policymakers, the lawyers. Everyone's really loud around the table, but we're not really listening to the public. And as we saw in some of the studies that have been put forth, there seems to be an openness to the public, an openness by the public and support towards moving forward with gene editing. That being said, I also don't think they're necessarily socialized enough on the aspect, and I don't think we're doing a good enough job of actually informing them to be able to have opinions.
There's huge debates about what it even means to consult the public, what it means to have public engagement in the governance of science. I would say one thing that ought to happen is that this should be, the dialogue should take place at all levels, including the political level. That is, people who make our elected representatives ought to know that it's a matter of concern to the public about what's happening with scientific technologies and their governance and their use. And so the dialogue should take place at all levels.
All right, so with that, I'd like to thank our speakers for being here and taking our questions. And I'm sure if there's more questions that haven't been asked, if you want to come down and talk to the speakers now, I'm sure they'd be open to that. We're just going to give them gifts to thank them for coming. And in general, we're very happy to have a big public turnout at these events. We think that the public opinion and the public's questions on these kinds of policy questions are important to have. And experts do like to get input from the public in terms of how they're going to bring this forward to further policy decisions. So we really thank you for your questions, and we thank you for being here, and we'll hope that you'll come to our next events. So thanks a lot.